Hello everyone, welcome to Sack Lunch News. Hello everybody, welcome to Sack Lunch News. Today's video is about gain-of-function research and the mRNA technology that's in the vaccines for COVID-19. Now you may be asking, 2014, the federal government halted funding gain-of-function research. They paused funding gain-of-function research. They didn't stop the actual research itself, they just stopped funding it. That's why Fauci went to DARPA to get the money to fund the gain-of-function research that he was going to do in the Wuhan laboratory. Because just because the US says you can't do it here and we're not going to give you the money to do it, doesn't mean you can't go somewhere else and get the money from somewhere else to do the same research. Think for a second. So, what is gain-of-function research? From virology.ws. Gain-of-function gives an organism a new property or enhances an existing one. The organism can be a virus, bacterium, fungus, rodent, bird, fish, well, really anything that can be experimentally manipulated. This part of the article is a bit shady, like the person is almost being purposefully misleading to a degree, maybe to make gain-of-function more palatable. Many have the impression that gain-of-function research involves making an organism more deadly. For example, increasing the capacity for a virus to cause disease. That impression is incorrect. Certainly gain-of-function might lead to a more dangerous organism, but most of the time that is not the goal. There are two broad approaches to gain-of-function research. In one approach, a virus is passaged in a host until a virus with different properties is obtained, like COVID-19 is in the human body. Another way to do gain-of-function research is to use recombinant DNA technology to engineer changes in the genome of the organism, which they did in the vaccines. I'll get into that here in a minute. An illustration of how gain-of-function research can be done on mice is the creation of a transgenic mice susceptible to poliovirus. Mice cannot be infected with the virus because they do not produce the cell receptor for polio. Basically what he's saying, in case you didn't catch that, he's saying they're taking a mouse and they're gonna make it susceptible to polio, because mice can't get polio because their body doesn't develop the receptors necessary to actually catch the disease. So check this out. They introduced the human poliovirus receptor gene into the mouse germline, leading to mice that produce poliovirus receptors. Generation down the line, 1 or 2 generations down the line, the mice will produce the receptors. After infection, these poliovirus transgenic mice can be infected and develop paralysis. These mice are a product of a gain-of-function experiment. Making something more deadly may not always be the goal, but from this person's own words, it can for sure be a goal, and that goal can be reached using gain-of-function research. So why did I bring up gain-of-function as the first part of this video? Because I want you to have an idea what it is as we dive into the vaccines and the spike protein causing adverse reactions. From planettoday.com. The lab origins of SARS-CoV-2 are coming clearer after scientists discovered a strand of DNA on the spike protein that matches a sequence patented by Moderna 3 years before the pandemic. This unique Moderna DNA was first filed for patent on February 4th, 2016. Now that's 2 years after the United States had paused funding gain-of-function research, and after the Wuhan laboratory had already been set up, and after Fauci and his teammate at Wuhan laboratory had went to DARPA to get the funds, but DARPA refused to give them the funds, so they ended up getting the funds another way, and then they still did all the bio research at the Wuhan laboratory. This DNA matches several letters of genetic code on the nucleotide and a genetic sequence on the unique furin cleavage site of the SARS-CoV-2 spike protein. This is the part of the virus that readily infects human cells. The likelihood of the code developing naturally is 1 in 3 trillion. That's why this was a plandemic. Moderna created and put a patent on a virus. You can call it a piece of the Great Reset if you want to. If you think this was bad, the vaccine is worse. Big Pharma hit us with a bioweapon twice. The first time to get you sick, the second time to keep you sick. What? You don't like what I said? Well, as Big Pharma would put it, you can't profit off the healthy. From blogginghounds.com, the messenger mRNA from Pfizer's COVID-19 vaccine is able to enter human liver cells and is converted into DNA, according to Swedish researchers at Lund University. Researchers found when the mRNA vaccine enters the liver, it triggers the cell's DNA inside the nucleus to increase production of line 1 gene expression to make mRNA. In that study, they present evidence that BNT162b2 mRNA is reverse transcribed intracellularly. That means it happens in your cells in your body, into DNA 6 hours after exposure, which the CDC said would not happen. Now, the CDC said the genetic material delivered by mRNA never enters your cell, stated on the CDC website titled Myths and Facts About COVID-19 Vaccines. The CDC also stated the vaccine does not interact or change your cells in any way. They said both mRNA and viral vector COVID-19 vaccines are discarded from the body once antibodies are produced. Pfizer never commented on the Swedish study, only stating the vaccine does not alter the human genome. Autoimmune disorders. Now, the Japanese study showed some of the mRNA moved from the injection site and through the bloodstream and were found in various organs such as the liver, spleen, adrenal glands, and ovaries 48 hours after injections. These tests were done on rats or lab rats. In another study, spike proteins were found on exosomes that mediate cell-to-cell communication for at least 4 months after the second dose was delivered. As long as the spike protein can be detected on the cell-derived membrane vessels, the immune system will be attacking the cells that release these vessels. Basically, your immune system attacks your organs. So, I'm not saying whether you should or shouldn't get the vaccine, but something cooked up in a biolab that can alter your DNA, I don't want any part of that. Now, you can go online and you can look all this up, and there's even more than this. This is just scratching at the surface, but I thought this would be a good first video to introduce myself to the COVID-19 scandemic, plandemic, great reset-ademic, whatever the fuck you want to call it, that's going on right now. See you in the next video. It appears Pfizer, Moderna, and the CDC lied about the COVID-19 vaccine, and it can alter your genetic code through your DNA genome.
피냐콜라다 아코세페 너의 피부색처럼 drip it 지금 내 핸드폰에 비친